Alright, welcome to the fifth set of notes in Chapter 8 over meiosis. We're going to be talking about the purpose of meiosis, which is the production of sperm and egg cells. This is a little bit different than mitosis, and we will be comparing the two. If you don't have a good handle on mitosis yet, please go back and review the screencast for those notes before you continue. So meiosis, as I said a moment ago, its purpose is the production of sex cells, which are also called gametes in animals and spores in plants. If you need to pause this slide to write everything down, please do. So let's revisit haploid versus diploid. We talked about these in our mitosis notes. Diploid, one of the things you need to be aware of is that you will see this designation of 2N for two sets of chromosomes. Diploid cells are made during mitosis. Haploid, or cells with only one set of chromosomes, are made during meiosis. And these are the sex cells, so sperm or eggs. So, you may have, hopefully I've given you in class, a foldable. These will go on the cover. Meiosis I goes on the left side, Meiosis II goes on the right hand side. If I haven't given you that yet, you can still take down these notes. Now, meiosis has two divisions, Meiosis I and Meiosis II. So we're going to go through prophase, metaphase, anaphase, and telophase, all during meiosis I. Then we're going to go through a second division, which we will call meiosis II, and it will be prophase II, metaphase II, anaphase II, and telophase II. There is also cytokinesis grouped in with telophase. So go ahead and note this is one key difference between mitosis and meiosis. Meiosis has two different cell divisions. And the thing that is vitally important to follow as we talk about the differences between mitosis and meiosis, it's all about the chromosomes. It's all about the chromosomes. Pay close attention to what's happening with the chromosomes. And having said that, we need to do a comparison here. So mitosis is on the left, meiosis is on the right. Mitosis makes diploid cells with two complete sets of their chromosomes. Meiosis makes haploid cells that have only one set of chromosomes in them. Mitosis makes our body cells, so everything that is not a sex cell. The daughter cells, which that's a term to use to refer to the new cells that are made, the daughter cells have two copies of every chromosome in mitosis, and they are identical to the parent cell. That's not true for meiosis. In meiosis, the daughter cells have one copy of every chromosome, and they are not identical to the parent cell or to each other. The new cells in mitosis are identical to the parent cell, and the new cells are different from the parent cell in meiosis. So here's a very good visual. These notes are available in a PDF form on my webpage, so if you would like this diagram, you can print it out. So here's mitosis with only one division. So you've got your duplicated chromosomes. Remember, they're X-shaped. During metaphase, they line up in the center of the cell. Anaphase and telophase, they get pulled to opposite ends of the cell, and you get two, and look at this, the 2N, you get two diploid cells. Meiosis is different. So here's meiosis I, and it walks you through, and then there's meiosis II, and we will walk through all of them. So here's the parent cell, and it has 6 chromosomes. Okay, so 2N. Here's the two longer ones. You could pair them up by size, just like we did on our karyotype. So these two, and these two. They duplicate for meiosis I, and they duplicate during S phase for prophase. They duplicate during S phase for meiosis as well. Now what happens in Prophase I? So this is the first division of meiosis. We actually, the mom and dad copies, so you see this if you want to pretend like the red ones are dad and the blue ones are mom. The mom and dad copies line up just like they would in a karyotype. So in your karyotype, when you placed the two chromosomes that were alike side by side, that's what's going on here. So your mom and dad chromosomes actually pair up. And we have a special name for that, and we'll go over those in a minute. And then you can follow the diagram on from there. We'll talk about each one of these in detail in the next few slides. So, mitosis prophase you should be familiar with. The sister chromatids form, so it condenses. The nuclear membrane breaks down. The centrosomes and the spindle fibers form. All of those things happen during prophase in mitosis. Now let's look at how meiosis is a little bit different. In Prophase I, the same things happen. So sister chromatids form, the nuclear membrane breaks down, centrosomes form, and spindle fibers form. Excuse me. The difference for meiosis in Prophase I is this thing called synapsis in tetrads. Go ahead and write this down. Synapsis occurs. And that's a process. So synapsis is a process that happens. Tetrads are formed and crossing over occurs. Crossing over is also a process, but tetrads are a physical thing. So let's talk about those. In prophase of mitosis, here on the left, the nuclear membrane breaks down, the spindle fibers start to form, and your chromosomes are just kind of floating around in there. Now look at the visual differences between mitosis and then meiosis I. Those mom and dad copies pair up. A tetrad is a mom and dad pair. Now notice they're called homologous chromosomes. These are the homologs. It's the same thing as the homologs that we talked about with the karyotype. So the chromosomes actually pair up according to their homologs, their homologous pair. Crossing over happens, and that's when your mom and dad chromosomes actually switch some DNA. Let's go into that a little bit more. So here we have dad is the pink copy, mom is the purple copy. Synapsis is when they pair up, and they actually do. Their little arms and legs stick together here. These sister chromatids, they actually do stick together. So the homologs get paired up in meiosis. This does not happen in mitosis. So our homologs pair up. Now this pair, and I've got the blue box around it, is called a tetrad. Think of 4 chromosomes, because, right, there's this copy from dad, this copy from dad, this copy from mom, and this one that's an exact copy of this one. So we've got 2 sister chromatids here, the purple one, this one is an exact copy of this one, and in our dad copy this is an exact copy of this one, right? And now the homologs have paired up. Again, you can see it here. Mom and dad copies have paired up. This is called synapsis. They're those homologous chromosomes, and the tetrad is formed. Now let's talk about the other thing that happens in Prophase I, and that is crossing over. So what is crossing over? Well, here's the definition, but pretty much your mom and dad copies, they've paired up, this is the tetrad, they're homologous, and they swap some DNA. They literally wrap their little legs around each other and swap. So you can see the diagram here, but an important question about crossing over, and one that you'll see a lot, is why does crossing over occur, and why is it important? And the answer to that is, see this chromosome here that is part orange and part red? That is an entirely new genetic combination of a chromosome. Entirely new. Which is part of the reason why you are so unique. You are a blend of your mom and dad, but more than just a blend of your mom and dad. The chromosomes that you got from your mom, because let's say you're going to get this one from your mom. If this one's from your mom, then that means the orange one was her dad and the red one was her mother. So there's your grandmother and your grandfather. So the DNA that you got from your mother, let's say you get this chromosome right here, this DNA that you got from your mother includes information from your grandparents, both of them. So you are entirely unique. You're not 50% your mom and 50% your dad. You're actually a little bit of your grandparents too. And this is important because it increases genetic diversity. And that's something that we'll talk about more when we get into DNA technology. Why is genetic diversity important? So let's move on. Metaphase. Mitosis metaphase. You remember this one? The sister chromatids line up in the middle of the cell. The difference here is the sister chromatids don't line up in the middle of the cell. The tetrads line up in the middle of the cell. So anaphase and mitosis, the sister chromatids separate, and you may have guessed by now that in meiosis, Anaphase I involves the tetrads separating, with one copy going to one side of the cell and the other copy going to the other side of the cell. Now, assuming that the red ones are dad and the blue ones are mom, notice that it's a mix of chromosomes. So not all of the ones from your mom go to one side and all of the ones from your dad go to the other side. That's not quite how it works. So, but there is one copy of, or one tetrad is split. So you have sister chromatids for this chromosome going to the left, this pair of sister chromatids going to the right. Alright, mitosis telophase. The nucleus reforms and you have two identical diploid cells with two copies of every chromosome. Meiosis telophase I is a little bit different. Two different haploid cells are formed, each with one copy of each chromosome. The cells are not identical anymore because they have split up the homologous pairs. So instead of splitting the sister chromatids, we have just split up the tetrads. We will split up the sister chromatids during meiosis II. So you can see that there are two cells that are formed in meiosis I, but they are still have their sister chromatids. Now, are there, and right now, there's one set of X's in each cell. So this one has the little bitty short one, and this one has the little bitty short one. This one has the middle one, this one has the middle one. This one has the longer one, and this one has the longer one. So we know that each cell has a complete set of the chromosomes, but it's still attached to its sister chromatid. In meiosis II, the purpose is to separate those sister chromatids. So let's take a look at that. Here are two cells that were formed in meiosis I. This one has the long chromosome, this one has the long chromosome. Has the short one and the short one. So here's Prophase II. In Prophase II, the spindle fibers attach to the center of the chromosomes. In Metaphase II, those are lined up in the center of the cell, similar to mitosis. And in Anaphase II, those sister chromatids are pulled apart. And in telophase and cytokinesis, you get 4 cells. These are the sex cells. One thing to notice as you look at these is that every one of them are unique. They have a different combination of genes. So even though this one has a short blue one and a short dark blue one, the longer one is different. This is why you and your siblings you may be similar, but you're not exactly the same, because crossing over happens differently each time, and it creates an entire new chromosome as far as genetics are concerned, a new combination of genes. So let's talk about this in a little more depth. In Prophase II, you have your sister chromatids, your X-shaped chromosomes here, and the spindle fibers attached to each one. In Metaphase II, you line those sister chromatids up in the center of the cell. Anaphase II, you pull those sister chromatids apart, and I'm showing two cells here because remember, these two were made during Meiosis I, so you have to include the two cells. And that's a good key. If you're looking at pictures and there are two cells undergoing the same process, it's a pretty good bet that it's Meiosis II. You can think of Meiosis II as in two cells. So if you see Anaphase II, there should be two cells going through anaphase. In telophase, you have 4 separate sex cells, and they are haploid. They have half of the original chromosomes. So what about this whole formation of gametes? Because we said the purpose of meiosis was to create gametes, or sex cells. In males, the process by which gametes are formed is known as spermatogenesis. In females, the process of growing and maturing an egg or an ovum is called oogenesis, or oogenesis, whichever you prefer. So, let's take a look at humans. If you look at the top, here's our original cell, and this is going to be the male. There are 46 chromosomes in human cells, so the first division of meiosis you divide those 46 into 23 and 23. Actually, I don't like this at all. I don't like this picture at all. Forget that. Actually, let's go back. Let's look at this picture. There are some problems with it because in this cell it says there are 46 chromosomes. And these 46 are duplicated during S phase, correct? So you would actually end up with 96 chromosomes. After the first division of meiosis, you end up with 46 chromosomes in each cell. After the second division, you end up with 23. So in this, there would be 46 in each of these after the first division. The same is true for the female. However, something interesting happens with the female. Because the egg, and here's the egg or the ovum, because the ovum has to contain all of the cell organelles that the baby is going to need. So all of the ribosomes, all of the endoplasmic reticulum, all of the Golgi apparatus, all of the mitochondria. Because the baby is going to need all of those when the sperm fertilizes the egg, and you can look, there is a huge size difference. The sperm just carrying DNA. There's no organelles in there to speak of. So the sperm cell's just carrying the DNA. The egg, on the other hand, has to carry all of the other cell parts and organelles. So it has to be as big or bigger than a regular cell. So what happens is all of the organelles from the original cell get shuffled to one side or the other. The chromosomes still get split just like they do for the male, except this one, because it is lacking all of the organelles, this one doesn't really function. It's called a polar body. It will divide again just because it's programmed to do that. But the important thing is that in meiosis, you produce 4 sperm cells and only 1 egg cell. So all of those organelles were shuffled to this cell. And then when it goes through meiosis II, the second division, all of those organelles are shuffled to this side again, leaving 3 what are called polar bodies that are non-functional. And these die and are broken down by the body. And nothing really happens with them. But they do contain the same number of DNA, the same amount of DNA as the other one. They just end up being broken down. And that's it.